glycine, which is important in cross-link in stabilizing the cross-links and maturing collagen fibers. So we have already discussed this in our previous topics, diba? The importance of hydroxyproline and hydroxylysine in collagen synthesis. So aside from being a tawag dito, being a composition of proteins, amino acids also serve as precursors of diverse biologic materials such as heme, purines, pyrimidines, hormones, neurotransmitters, and biologically active peptides. So, proteins talaga is abundant in our body. So, like for example, the histamine, they play a role in many allergic reactions as well as mga neurotransmitters. So examples are the serotonin, dopamine, norepinephrine, and epinephrine. And the drugs that are being given to patients who have neurologic and psychiatric conditions perform or these drugs hit these conditions by altering the metabolism of these neurotransmitters. So again, we will be considering each of the amino acid and their functions or in being a specialized protein. So we will start with the alpha amino acids. So for alanine, they serve as, we already know this, they serve as a carrier of ammonia and the carbons of pyruvate from skeletal muscles to liver via the Cori cycle. So together with glycine, they constitute a major fraction of the free amino acids in the plasma. So, ayun, so glycine and alanine. Then we have the arginine. So for arginine naman, di ba, going back to the urea cycle, they serve as carriers of nitrogen atoms in urea biosynthesis. So what happens is the guanidino group of arginine is incorporated into creatine. And also here, creatine. So arginine, it can contribute its nitrogen to form urea. Also, its guanidino group can be incorporated into creatine. So here. And also following conversion to ornithine, so arginine becomes ornithine, its carbon skeleton becomes that of the polyamines tyrosine and spermine. Here. So, maraming contribution ang amino acid na arginine. So, the reaction is catalyzed by nitric oxide synthase, which is a 5-electron oxidoreductase with multiple cofactors and converts one nitrogen of the guanidine group of arginine to L-ornithine and nitric oxide, which is an intercellular signaling molecule that serves as a neurotransmitter, smooth muscle relaxant, and vasodilator. So this was already mentioned before na arginine can help in our diet, diba? So they can help in vasodilation because of the nitric oxide. Then we have another alpha amino acid, cysteine. So for cysteine, they participate in the biosynthesis of coenzyme A here by reacting with pantothenate to form 4-phosphopantothenol cysteine. So this one, so cysteine reacts with pantothenate and becoming 4-phosphopantothenol cysteine. Okay. Then 3 enzyme catalyzed reactions convert cysteine to taurine. So this one, so cysteine can be converted to taurine using these 3 enzymes. And this can displace the coenzyme AYP of choline-CoA to form the bile acid taurocholic acid. Also, conversion of cysteine to taurine is initiated by its oxidation to sulfinoalanine. So initially, here, so it becomes sulfinoalanine, and this is catalyzed by the non-heme iron enzyme cysteine dioxygenase. So that is the reaction. And after that, the carboxylation of cysteine sulfonate by sulfonylalanine decarboxylase forms hypotaurine, whose oxidation by hypotaurine dehydrogenase forms taurine. So that was just the reaction on how cysteine becomes taurine. So it's a 3-enzyme process. Another one is for glycine. So many metabolites and pharmaceuticals are excreted as water-soluble glycine conjugates. So glycolic acid and hippuric acid formed from the food additive benzoate. So here, benzoate, and this becomes hippurate or hippuric acid. So many drugs, drug metabolites, and other compounds with carboxyl group are conjugated with glycine, which makes them more water soluble. And when they become water soluble, so they are excreted in the urine. Glycine is also incorporated into creatine. The nitrogen and alpha carbon are incorporated also into the pyrrole rings and the methylene bridge carbon of heme. So the entire glycine molecule becomes also the atoms 4, 5, and 7 of the purine. So as you can see, there are many functions or specialized functions of the amino acid glycine. For histidine naman, so this is histidine, the carboxylation of histidine to histamine is catalyzed by pyridoxal 5-phosphate dependent enzyme, histidine decarboxylase. So histidine, this becomes histamine, yung important for mga allergic reactions. So histamine is a biogenic amine that functions in allergic reactions and gastric secretion, and they are present in all tissues. So in the hypothalamus, so concentration in the brain hypothalamus, this varies in accordance with circadian rhythm. So yun, histidine helps in forming histamine. So histidine compounds present in the body includes carnosine and dietary-derived ergothioneine and anserine. Ayun, so this one. Ergothioneine, carnosine, and anserine. And the carnosine and homocarnosine, their precise physiological functions are unknown, but it is said that they are the major constituents of excitable tissues, the brain and the skeletal muscles. And we have here a condition, the Wilson disease, wherein in this condition, the urinary levels of 3-methylhistidine are unusually low. So Wilson's disease. Then for methionine, the major non-protein fate of methionine is conversion to S-adenosyl methionine. And this is the principal source of methyl groups in the body. So yun ang major non-protein fate ng methionine. And biosynthesis of S-adenosyl methionine from methionine and ATP is catalyzed by the enzyme methionine adenosyl transferase. So that's the enzyme involved. And in the human tissues, we have 3-MAT or methionine adenosyl transferase or MAT isozyme. So we have MAT1 and MAT3 found in the liver and MAT2 in non-hepatic tissues. So there's here also a condition, hypermethionine, which results from severely decreased hepatic MAT1 and MAT3 activity. However, if there is residual MAT1 or MAT3 activity and the MAT2 is normal, a high tissue concentration of methionine will ensure synthesis of adequate amounts of S-adenosyl methionine. So, ayun. So, kahit residual activity lang of MAT1 or MAT3 activity and MAT2 is normal. So, we can still form adequate amounts of S-adenosyl methionine. So, here, this is how spermine and spermidine is formed. So from the carboxylation of S-adenosyl methionine by methionine decarboxylase, 3-carbon atom and the alpha amino group of methionine so contributes to the biosynthesis of polyamines, spermine and spermidine. So this is also from methionine. These polyamines function in cell proliferation and growth So there are growth factors for cultured mammalian cells and stabilized intact cells, subcellular organelles, and membranes. Pharmacologic doses of these polyamines are actually hypothermic and hypotensive. And polyamines also bear multiple positive charges and are readily associated with DNA and RNA. So, in other words, methionine is important with DNA and RNA. So here is the catabolism of, this shows the catabolism of polyamines. So how aspartame eventually becomes tyrosine and ammonia and carbon dioxide. So next amino acid is serine. So serine participates in the biosynthesis of sphingosine and of purines and pyrimidine. So they provide carbons 2 and 8 of purines and the methyl group of thymine. So we have a condition, so we already know this homocystinuria. So this is a genetic defect in cystathionine B synthase that catalyzes the pyridoxal P-phosphate dependent condensation of serine with homocysteine to form cystathione. So if there is a defect in the enzyme, so there is homocystinuria that occurs. Then we have tryptophan. So for tryptophan, initially, so there is hydroxylation of tryptophan to 5-hydroxytryptophan by liver using the enzyme tryptophan hydroxylase, and after which there is decarboxylation, which forms serotonin. So serotonin, this one, or another name is 5-hydroxytryptamine. And this serotonin is a potent vasoconstrictor and stimulator of smooth muscle contraction. So tryptophan synthesizes or becomes eventually serotonin. And catabolism of this serotonin is initiated by deamination to 5-hydroxyindole 3 acetate, and this is catalyzed by MAO or monoamine oxidase. So it becomes this one, 5-hydroxyindole 3 acetate. And ayun. So the cyclic stimulation that follows administration of iproniacid results from its ability to prolong the action of serotonin by inhibiting monoamine oxidase. So this iproniacid, so its function is this inhibits here the monoamine oxidase. So what happens is there is prolongation of the action of serotonin. So there is a condition in carcinoid or argentophenoma tumor cells. Here, the tumor cells overproduce serotonin. So the urinary metabolites of serotonin in patients with carcinoid include N-acetylserotonin, glucuronide, and the glycine conjugate of 5-hydroxyindole acetate. Okay, so this is a condition with an overproduction of serotonin. So serotonin and 5-methoxytryptamine are metabolized by the corresponding to the corresponding acids by monoamine oxidase. So here, serotonin and this one is also acted upon by monoamine oxidase, and they become their corresponding acid. So N-acetylation of serotonin followed by its own methylation in the pineal body forms melatonin. So yung serotonin, pwede dito siya mag-act or here and here. This one reaction forming melatonin. Circulating melatonin is taken up by all tissues, including the brain, but is rapidly metabolized by hydroxylation, followed by conjugation with sulfate or with glucuronic acid. So those are the important things for tryptophan that the amino acid tryptophan contributes. So kidney tissue, liver tissue, and fecal bacteria all convert tryptophan to tryptamine, then to indole 3 acetate. So principal normal urinary catabolites of tryptophan are 5-hydroxyindole acetate and indole 3 acetate. So these are the catabolites for in tryptophan catabolism. Then we have tyrosine. So this is again another important amino acid because neural cells convert this tyrosine to epinephrine and norepinephrine. So this starts with the DOPA decarboxylase. This one, which is a pyridoxal phosphate dependent enzyme, forms dopamine. So tyrosine becomes dopamine using this enzyme. Then subsequent hydroxylation catalyzed by dopamine beta oxidase then forms norepinephrine. So very important ang amino acid na tyrosine. So in the adrenal medulla, phenylethanolamine and methyltransferase utilizes S-adenosyl methionine to methylate the primary amine of norepinephrine to become epinephrine. So this one, this uses up S-adenosyl methionine with the help of this enzyme to form epinephrine. So this occurs in the adrenal medulla. So DOPA is also an intermediate in the formation of melanin in melanocytes. And here, DOPA is actually here, this one, before it becomes dopamine. So DOPA also is an intermediate in the formation of melanin. So tyrosine, aside from becoming dopamine, epinephrine, and norepinephrine, they're also precursor of triiodothyronine and thyroxine, so in the thyroid. So also a very important amino acid. Then we have phosphosylene, phosphothreonine, and phosphotyrosine. So phosphorylation and dephosphorylation of specific serial, threonyl, or tyrosyl residues of proteins regulate the activity of certain enzymes of lipid and carbohydrate metabolism and of proteins that participate in signal transduction cascades. So all this amino acid is important in the metabolism of both the enzymes needed in lipid and carbohydrate metabolism. Then we have sarcosine. So sarcosine or N-methylglycine. So the biosynthesis of this one is biosynthesis and catabolism occurs actually in mitochondria. So the formation of sarcosine from dimethylglycine is catalyzed by flavoprotein, dimethylglycine dehydrogenase, and this requires reduced terol pentaglutamate, so TPG. Ayun, so, and this forms now sarcosine. So traces of sarcosine can also arise by methylation of glycine. So using this reaction, so glycine is adenosyl methionine. So again, the formation of sarcosine. So catabolism of sarcosine to glycine also requires reduced terol pentaglutamate. So this is the breakdown naman of sarcosine, which is catalyzed by flavoprotein sarcosine dehydrogenase, also requires this TPG. So it becomes glycine. The methylation reactions that form and degrade sarcosine represents important sources of one carbon. And the FADH2 is reoxidized via the electron transport chain. So we have also the creation of creatine and creatinine. So creatinine is formed in muscles from creatine phosphate by irreversible non-enzymatic dehydration and loss of phosphate. So creatinine. And since the 24-hour urinary excretion of creatine is proportionate to muscle mass, it provides a measure of whether a complete 24-hour urine specimen has been collected. So later on, if during your third year, if you already have your OB or OB rotations, you will learn that in determining yung mga patients with preeclampsia, so ito yung ginagamit namin to determine the creatinine clearance. So the 24-hour urinary excretion of creatinine, because this is proportionate to the muscle mass of the body. So ayun, so that's the use of this 24-hour urinary excretion. So these amino acids, these three actually participates in creatine biosynthesis. So we have glycine, arginine, and methionine. And synthesis of creatine is completed by methylation of guanido acetate by S-adenosyl methionine. So this one, guanido acetate. So ayun, so and with S-adenosyl methylation of S-adenosyl methionine, so they form creatine. Okay, so that is the last for the alpha amino acid. So now we are going to the non-alpha amino acid. So this includes beta-alanine and beta-amino aldehyde. So these two are formed during catabolism of the pyrimidines, uracil, and thymine. So beta-alanine formed from uracil and amino isobutyrate from thymine. So traces of beta-alanine also results from the hydrolysis of beta-alanyl dipeptides by the enzyme carnosinase. Beta-amino isobutyrate also arises from transamination of methyl malonate semialdehyde, which is a catabolite of L-valine. So for this one, mga non-alpha amino acid actually, there are only three of this. So this one, this is the creation of succinyl-CoA. So there is an initial reaction of beta-alanine catabolism. The initial reaction of beta-alanine catabolism is transamination to malonate semialdehyde. So, ayun, malonate semialdehyde and subsequent transfer of coenzyme A from succinyl-CoA forms malonyl-CoA semialdehyde. And then oxidized to manyl-CoA and decarboxylated to the amphibolic intermediate acetyl-CoA. So the transamination forms methyl malonate semialdehyde, which is converted to amphibolic intermediate succinyl-CoA. So it's again another intermediate. So disorders of metabolism arise from defects in enzymes of the pyrimidine catabolic pathway. So disorders in the metabolism of these two non-alpha amino acid happens if there is a defect in the enzyme of the pyrimidine catabolic pathways. So principal among these are disorders that result from a total or partial deficiency of dihydropyrimidine dehydrogenase. So if there is a problem with this enzyme, so most disorders arise if there is a problem with this dihydropyrimidine dehydrogenase. So this is another non-alpha amino acid, the beta-alanyl dipeptide. So this includes carnosine and serine, which activates myosin ATPase, chelate copper, and enhance copper uptake. So beta-alanine imidazole buffers the pH of anaerobically contracting skeletal muscles. So ayun ang one function. So biosynthesis of carnosine is catalyzed by carnosine synthase in a two-stage process. So so biosynthesis of carnosine is catalyzed by carnosine synthase in a two-stage reaction that involves initial formation of an enzyme-bound acyl-adenylate of beta-alanine and subsequent transfer of the beta-alanyl moiety to L-histidine. So here is the formation of the carnosine. So hydrolysis of carnosine to beta-alanine and L-histidine is catalyzed by the enzyme carnosinase. And there is a condition, carnosinuria, which is a heritable disorder of carnosinase deficiency. So if there is a deficiency with this enzyme, this can lead to carnosinuria. Okay, so homocarnosine, which is present in human brain at higher levels than carnosine. So there is a homocarnosine. So this is actually present in our brains, and it is in a higher level than carnosine. So they are synthesized in brain tissue by carnosine synthase. And this homocarnosine is not hydrolyzed by serum carnosinase. So this carnosinase can only catabolize the carnosine and not the homocarnosine. So there is also a rare genetic disorder, homocarnosinosis, and this is associated with progressive spastic paraplegia and mental retardation. So ayun, carnosinuria and homocarnosinosis. Then we have this one. So this is also another important non-alpha amino acid, so the GABA, the gamma aminobutyrate. So they function in brain tissue as an inhibitory neurotransmitter by altering transmembrane potential differences. So this is formed by the carboxylation of glutamate by L-glutamate decarboxylase. L-glutamate decarboxylase from glutamate becomes aminobutyrate. And ayun, then transamination of gamma aminobutyrate forms succinate semialdehyde, which can be reduced to succinate semialdehyde, which can be reduced to hydroxybutyrate by L-lactate dehydrogenase, or be oxidized to succinate and uh, hence via the citric acid cycle to CO2 and water. So ayun, oxidized here and becomes succinate. Okay, so that is for the GABA. So if there is a defect in GABA or aminobutyrate, gamma aminobutyrate aminotransferase, so this can lead to a rare genetic disorder of GABA metabolism, and in enzymes that participates in the catabolism of the GABA subsequent to its postsynaptic release in the brain tissue. So this aminotransferase is actually an enzyme that participates in the catabolism of GABA before subsequent to its postsynaptic release in brain. So ayun, so there is a problem in the breakdown or metabolism of GABA. Then we have also the 4-hydroxybutyric aciduria, wherein there's defects in succinic semialdehyde dehydrogenase. This is again a rare metabolic disorder of the GABA aminobutyrate of the GABA or the gamma aminobutyrate metabolism, and this is characterized by the presence of 4-hydroxybutyrate in urine, plasma, and cerebrospinal fluid. So as of the present, there is still no available treatment for the accompanying mild to severe neurologic symptoms in this condition. Okay, so ayun, basically this is a short chapter. So in summary lang, so in addition to serving, diba, we all know na that amino acid is the structural unit of protein. So in addition to that, in serving structural and functional roles in proteins, alpha amino acid participate in a wide variety of other biosynthetic processes, diba? So we have seen na pwede siya sa ito sa epinephrine or epinephrine. So, mga neurotransmitters, di ba? So, in creatinine. Ayun. So, arginine provides the formamidine group of creatine and the nitrogen of nitric oxide. So, we also know this na so, role ng arginine. So, they provide its formamidine group becomes creatinine, and its nitrogen group becomes nitric oxide, which is a vasodilator. So via ornithine, arginine provides the skeleton of the polyamines tyrosine, spermine, and spermidine. So, ayun, marami din siyang function. So cysteine provides the thioethanolamine portion of coenzyme A, and following its conversion to taurine is part of the bile acid taurocholic acid. Yun, so that's from cysteine. So glycine naman participates in the biosynthesis of heme, purines, creatinine, and N-methylglycine or the Tyrosine. And many drugs and drug metabolites are excreted as glycine conjugates because ito siya, glycine conjugates are increasingly are water soluble, so they can be excreted in the urine. So the carboxylation of histidine forms the neurotransmitter histamine. So histidine becomes histamine. Histidine compounds present in the human body includes ergothioneine, carnosine, and serine. Ayun, so these three. Then S-adenosyl methionine, which is the principal source of methyl groups in metabolism, contributes its carbon skeleton to the biosynthesis of the polyamines spermine and spermidine. So S-adenosyl methionine, ayun, spermine and spermidine. So in addition to its roles in phospholipid and sphingosine biosynthesis, the serine provides carbon 2 and 8 of purines and the methyl group of thymine. So this is another role of serine. So they provide the carbons of purine. So tryptophan naman. So key tryptophan metabolites includes, we know, tryptophan can become serotonin, and further on in the process can give rise to melatonin. So kidney and liver tissue and also fecal bacteria convert tryptophan to tryptamine and hence to indole-3 acetate. So the principal tryptophan catabolite in urine are indole-3 acetate and 5-hydroxyindole acetate. Tyrosine forms norepinephrine and epinephrine. So yes, we have already shown this reaction. So it can become norepinephrine and then epinephrine, and then can also give rise to the thyroid hormones triiodothyronine and thyroxine. So also the enzyme catalyzed interconversion of the phospho and the phosphoforms of peptide-bound serine, threonine, and tyrosine plays key roles in metabolic regulation, including signal transduction. Then glycine, arginine, and S-adenosyl methionine all participate in the biosynthesis of creatine, which as creatine phosphate serves the major energy reserve in the muscles and brain. So glycine, arginine, is adenosyl methionine. So excretion in the urine of its catabolite, creatinine, is proportionate to muscle mass. Okay, so we also have mentioned that. Then beta-alanine, so this is now the non-alpha amino acid, and beta-amino isobutyrate, both are present in tissues as free amino acid. So beta-alanine also occurs in bound form in coenzyme A, carnosine, and serine, and homocarnosine. And catabolism of this involves a stepwise conversion to acetyl-CoA. So analogous reactions catabolize beta-amino isobutyrate to succinyl-CoA. So catabolism ng alanine, beta-alanine, acetyl-CoA, and catabolism ng amino isobutyrate, succinyl-CoA. And disorders of beta-alanine and beta-amino isobutyrate metabolism arise if there are defects in the enzymes of pyrimidine catabolism. So we have also mentioned that, and the carboxylation of glutamate forms yun, glutamate formation of GABA, so gamma-aminobutyrate. So there are two rare metabolic disorders that are associated with defects in GABA catabolism. Yun na pala ang last. Okay, so so far yun ang ano natin, specialized proteins that are associated with some of our important amino acids in the body. Okay. So basically, this is a short chapter na lang, and this will be included in your quiz tomorrow, along with our previous topic last week.